Hi, my name is Resa Nelson. I'm from Hofstra University in Long Island, New York, and I'm a bio major, biochemistry minor with a cell bio. This summer, I'm working in the Flanagan lab with Laura to investigate when you knock out the MGAT5 gene, the cortical thickness and the cortical composition of the brain, the physiology is very different in embryonic mice. So we want to see if that difference is maintained in adulthood with 3-month mice. I like how involved it is and like how many different steps there are to achieving this one task. So from start to finish, we have to do a lot of perfusions, we have to do sectioning, staining, imaging. I like to see that pipeline from beginning to end, which is just amazing. I'm most excited to see the results, really, because you get to read the papers. The lab knows who wrote the paper, but I don't. So to be able to see the start of the embryonic mice and then to see the adult mice, I think would be really cool, just to see what the answer is and to figure it out. We've been watching a lot of movies, just going to lunch and dinner, and just hanging out with the cohort, 20 amazing people. I really enjoy being with them. One highlight of the summer so far was the 4th of July. We got to go on the carnival rides, the Ferris wheel, we went to the trolley, had some good food. I just like being at the beach. It's just, I will always remember that day.
Hello, my name is Laura Tennis. I am a 5th year MD PhD student here at UCI, and I am in the Department of Anatomy and Neurobiology, and I am this year's mentor for Resa Nelson. In our lab, one of the major goals is to understand the factors that influence neural stem cell differentiation, so whether a neural stem cell becomes either the communicating cells in the brain, neurons, or astrocytes, the support cells. And what we've done is previous work in the developmental sphere to look at how cell surface triggers influence that process. And so what Resa's project does is build on that work, where we've identified a gene called MGAT5 that is important both for neural stem cell differentiation as well as early brain development. And it looks at whether some deficiencies we observed in a knockout mouse for MGAT5 persist into adulthood, or whether the brain is able to repair those deficits. In regards to mentorship, it's been such a joy to work with Resa. She comes with her own experiences, both in confocal and tissue culture, but she's also been able to expand on the work that she's done previously by continuing to build her microscopy experience, her staining experience, and as well develop as a researcher through both the CNLM program and all the opportunities that they offer, but as well here talking with all the amazing researchers in the lab and getting the perspectives on graduate school and as well research. And I think we all really enjoy having undergraduates in the lab. I think it helps keep the spirit of the lab high and also helps us support the next generation of scientists. One thing that I really enjoyed about working with Resa is she's very detail-oriented, and she asks questions that kind of highlight her curiosity and allow me to expand my own knowledge. In a day-to-day, you don't always think about the little steps in a protocol, and when someone's asking very specific questions, it helps me find gaps in my knowledge and push my knowledge further. So I really have loved working with Resa and other undergrads in the lab for that reason, and I'm so excited to see her continue growing within the lab.